I'm good. Cool. We'll keep this speed then. Looks pretty good to me. Taylor Ann, we'll just call this the start of Transect 11.
Yep.
For World Octopus Day, our crowdsourcing, we figured out that it is about 3 hours and 50 minutes back in time if you want to see the flapjack octopus from this morning. So that was posted a couple minutes ago. Thank you for whoever helped us out with that. Another person said it was around 7:45 a.m. Pacific Daylight Time.
Awesome. Sounds like we have some committed folks joining us. Been watching for a long time. So Erica, we've had some fun here getting some questions and comments in from the public. So that's
great. I noticed we had a crab hitchhiker. We were chuckling on our daily debrief.
Oh, it's still here. It just poked in from the port side a moment ago.
Oh, good.
As long as it doesn't completely obscure the view while transecting.
Here's another question that's kind of fun. Have you seen any marine worm species on this dive? I know on the last watch we were looking at some, looked like tube worms growing on a tanner crab, but we've seen maybe some feathery feeding appendages from some maybe polychaete worms as well. But any thoughts on that, Erica or Kirsten?
We have seen some tube worms. We haven't done a whole lot of close-up shots on the sediment to see many of them, but they are definitely in the sediment. Whole variety. There are polychaetes. There are acorn worms, maybe a little deeper. And if we see some, we'll point them out because they're quite spectacular when they come out and they create some trails on the seafloor. Kirsten, have you seen any worms?
No, just, lots of homes for possible worms in the soft sediment we talked about.
And another person asked, how long are these dives typically?
There's no real typical. Why don't you go ahead, Erica?
Oh, well, the one we're on now is about, 24 hours. So the ROV pilots might want to comment on that, or maybe Taylor Ann, the data logger, but we tend to stay down for a long period of time, 24/7, because there is a lot of up and down time. It takes about an hour to get down to about 1,000 meters. Correct me if I'm wrong, and then a little longer to get back up. And then the ROV needs 4 hours on deck to do a turnaround and maintenance and make sure everything's working well. So feel free to add to my novice discussion on that.
Yeah, so it just depends on the goals of the dives. So we've had dives last maybe 8 hours if it's something where we're not going too deep and we have our goals planned out. But I've also been on dives that have been up to like 36 hours. I'm not sure if they go beyond that or not. But yeah, it ranges depending on the goals of the dive and what we're trying to do. But yeah, our last 2 dives have been either 20 or 24 hours. And we switch watch teams every 4 hours so that people get a chance to sleep and eat and process all of the data that we're collecting. And it's pretty fun. So I'm on the 12 to 4 watch, so that's AM and PM.
Great, and we've had 3 or 4 questions about how do the animals react to light since they're not used to seeing bright light?
Yeah, so some organisms, some fish may be attracted to it. It may seem like a disturbance to them. And scavengers like grenadier may actually be attracted to the commotion. And I don't know if it's the lights or the hum of the thrusters, but yeah. And then there are some organisms that will not want to be around the light. They have low light capabilities and they may get blinded by the light actually, because it's by the makeup of their eye structure. So both sides of the coin. Some attracted, some don't want to be near it.
But it is interesting to think about the fact that even though sunlight doesn't make it down to this depth, a lot of these species are bioluminescing. So there are, if we were to turn all the lights off, it wouldn't, there would be spots, flashes of light around. So, and those lights, like Erica was saying, can be used to distract or attract things, confuse predators, draw in prey. So some species may use that light a little bit in that capacity. But as you see, we go by, most things don't seem to move out of the way. Though of course others may have moved before we get there.
Thank you. Just going to give a quick shout out to third graders in Davenport, California, are representing. They are watching the program today as kind of like a virtual field trip. They are going to the deep sea at Pacific Elementary School. Feel free to put in a question, Pacific Elementary, if you want to ask any of our scientists or our technicians and pilots.
So a reminder for anyone who's just joining us, we are currently conducting a transect. So we are collecting video data in a standardized way as we transit across the seafloor bottom. So although it may look like we're not doing anything, we are doing a lot.
Yeah, and this is Erica. I'm the watch lead during the shift, and I just came back from a daily debrief call to just plan for this dive and see where we are. So some of the things we're going to be targeting, our goals for this next 4 hours is to run some more time transects, as Kirsten said, in all different habitats, both soft, hard, and mixed. We also want to keep our eyes out for any sponges that we may want to collect. We have a running list of sponges of interest for taxonomists back in their institutions, including the pipe cleaner type looking predatory sponge Asbestopluma. And then if we see any large, round, broad, low base sponges that may have a fish inside it, we will probably want to do a 3D mosaic or a 360 of it. And then we have one last push core for the rest of the whole dive. We have one left, so we'll probably be using that for either near a bunch of corals where there's high diversity or lots of sea pens. Right now, this sediment may be a thin veneer, so we'll want to make sure that it really is a deep, soft sediment when we take that core. And then we have one Niskin bottle left, and we will determine where we want to place that. Some of our priority areas would be in a place where there's a diverse hard substrate area with lots of coral diversity, or if we collect something truly unusual, we'll want to collect a water sample, and that'll be used for eDNA studies. So looking at animals will slough off their tissue or their scales, and it gets left in the water column for a few days, and we can actually determine what animals have been there, even though we may not have seen them. And it's great to be able to match up that eDNA to what we are seeing and maybe things we aren't even seeing, but they're showing up in the water. So that's what we've got planned for the next 4 hours and for the rest of the dive. It'll end later tonight. Just want to give you a briefing. So feel free to answer any more questions, and we're going to set up for another transect soon. Oh, no, wrong one right now, aren't we? Yes, we're on one.
Yes, we are on one, yeah. Looks like a
Great.
gopher sole we are passing and a thorny head.
And Kirsten, people were asking why the thorny heads and some of the other fish are so sedentary, why they're not moving around that much. Any thoughts on that?
They tend to be in the oxygen minimum zone. So there is a lot less oxygen available for them to use to expend energy. And there are some scientists on the science chat, so feel free to provide some answers and we'll translate. But they may be saving their energy for actually capturing prey rather than just swimming around and using up that energy otherwise.
I also like to ask the questions that I'm trying to ask as many as possible. Thanks everyone for your patience. They're flying in. This question I had too. When the kelp got stuck in the slurp yesterday, when there was some kelp sampling, did you manage to get it through? Did you manage to get it back up? Were you able to clear everything out? Anybody happen to know that?
It is still in the hose.
Actually, they did free it. They did
Oh,
get
they
it
did?
out. Yeah, I guess it happened on the last watch
Okay.
when they were collecting something else. And they also collected another piece of kelp successfully. So we now have a piece of kelp in the flesh jar and in our sample jar number 1, I believe.
Wow.
Yeah.
That thing was really stuck in there. I'm surprised they got it out.
Okay, that's
I
the
love that we
end
have
of our 150-meter transect. So mark that.
The transect.
T11E.
Thank you.
So we're still on our way to 2T. Let me know if anything changed from that. And then when we're at 2T, I'll head over to 2V.
Sounds good.
TU is a bit downslope. I don't think we should head there first. Seems like V would be. Well, let's take a look. Maybe you can help me decide here.
Okay, I appreciate that feedback. That's great.
Sure. So I have it up on HiPAC, which I think you're seeing on channel 3. We are still heading towards 2T, which is over here. And then, so this section right here, that's a bit jumbly. I took a look at the multi-beam data, and I believe that that's artifact. So I think it's mostly a smooth slope here, but there's, I don't, we'll see. We may find that it's interesting, but I think that this is unartifacted. And then, so we could go, the problem is it's quite a bit downslope to go to 2U, or we could go to 2V, try to backtrack to 2U, and go up to 2W, or we could drop down a bit and kind of cross through this section here, through that bit that I believe is artifact.
Yeah, it might be good just to visit real quickly.
Sure.
You know, just to peek at it and see what it looks like and see if it really is an artifact.
Mm-hmm.
But like you said, we don't want to be going down slope too long. So whatever you think is the best way traveled would be nice.
Sure. So once we get to 2T, maybe I'll try to bisect this, the point between TU and TV, and then we can decide if we're going south to TU or north to 2V.
Sounds good.
Okay.
Erica and Kirsten, are we open for some more questions? I was trying to give the navigation and decision-making some space there.
Yeah, I think so. We're going to just traverse a little bit till we get past 100 meters.
Great. Another question is, can you use the ROV arm to make sure the soft ground is deep enough before doing a push core?
Definitely. That is sometimes the trick.
It's not a backhoe. Manipulator.
What I'm hearing is it's kind of a hack. Like, this is a precision instrument. You don't want to just be dipping it in the sediment, even though that may give you some information. All right, on a slightly different note, have we seen any deep sea fish spawning?
We have not that I know of. There have been observations of some fishes that actually brood their eggs, like the blob scalpin can actually brood their eggs. There've been observations up at the Mendocino Ridge where they have countless number of eggs that they actually sit on or close by until they hatch. But on our dive so far, we have not seen any fish spawning.
Great, and this is back to the ROV. Can we get an overview of all the tools that the ROV has?
Well, there's a lot of them. Specifically, it has 2 manipulator arms, about 13 lights and 7 cameras, 1 high definition camera. It's got a couple bio boxes for samples, a slurp hose, and some slurp buckets. It's got a couple of different sensors for sensing oxygen, temperature. There's a whole bunch of stuff. Could keep going for a long time. Bunch of sensors to tell how the ROV is moving, to communicate with the ship to tell its position, the USBL. Those work using a combination of underwater acoustics and there's some motion instruments like a gyroscope to tell how the ROV moves. And yeah, I don't know, that's a quick rundown of some of the capabilities, instruments on the ROVs.
That's great. Thank you so much. We had a question
Justin,
about whether...
Can I circle back to the fish eggs? Tom Leidig from NOAA Fisheries reminded me that we have seen some skate egg cases and cat shark egg cases on these dives the last couple of days. And some of them tend to have these long tendrils that sometimes will attach the skates, or the cat sharks will put them near rocky habitat or near corals. And I've seen some of the tendrils actually wrapped around some of the structure to keep them there. And so yeah, we have seen those sorts of egg cases, and those are a little more common in the deep sea. We'll see more of those here and there. So we'll be on the lookout. If we see any, we'll point them out.
Thank you, Erica and Tom. Oh, here's a good question. Are you all on the boat, or are you able to remote in? Well, I hope somebody's on the boat. I guess maybe we can give brief introductions and say whether we're on the boat or not. I'll start. I'm Justin, and I'm working for Greater Farallones on the education team. I am remote. I am ashore, beaming in from San Francisco. Kirsten, do you want to go next?
Oh, sure. I am also in California right now. I was supposed to be on the boat. Justin, were you originally going to be on the boat or calling in?
Yes, I was.
Yes, okay. Yeah. So, yes, I am at home and hope to be back out on the ship next year.
Erica, you want to go next?
And yeah, and I think the question, I was busy doing some data. I think the question was, are we, the scientists, at home? Is that right? Is that the question?
Or on the boat, yeah. Just like who's at home, who's on the boat?
Yeah, so with COVID, a lot of us have been teleworking anyway. So we have sort of set up our computers at home, and now with the ramping up this cruise, we have tried to duplicate as much as we can.
All right, so we're going to alter course now to 110. It's going to be a bit downslope, but hopefully not too downslope. But that's the end goal, is down in that direction.
Sorry,
so I'm going to call that in now. Argus will kind of loop around that way, and you can kind of crab along.
Okay.
Bridge nav. Yes, can we step 150 meters, bearing 110, speed 0.3? And can you check your microphones? I think one of them's on. Thank you.
Sorry, just as I was explaining all the setup at home, I lost my audio. So I was saying that we've tried to duplicate as much as we can what we can see when we're actually on the ship. So many of us have maybe 2 or 3 video screens up in our homes office, delivering all the video feed, the data feed, and the audio feed, as well as our supporting map documents and cruise plans. So yes, I am home.
You have a target up ahead of you. Maybe we can get there. Might just be a fish. We'll see if it's persistent.
Oh yeah.
So we've been passing a couple of Dover sole and a mixture of short spine and long spine thorny heads.
Coming up 10 meters out.
And Rennie, I wanted to circle back with you. Our USGS scientist on shore did confirm what you said in that the data around 2U is poor data.
Yes.
So we may not need to go down there, just like you thought.
Okay, so I'm still choosing, I can show you here. We're going to go slightly along contour / a little bit downslope, unless you want to skip 2U completely and go to 2V. But for now, we're kind of threading the needle a little bit between the two on this next
Yeah,
transect.
that's
All right, there's
great.
our target. There's we saw that on the sonar.
Yeah.
Yeah. No, I like your plan. That's good.
Okay.
What
What do we
is
see
this?
up here?
Whoa.
We are east of a old Navy military dump site, Navy dump site. So this may be part of it or something different altogether.
It's a reminder that the 2 green
Yeah,
lasers are 10 centimeters apart. So we are currently transiting between transects. So we are trying to cover at least 100 meters before we consider starting another transect so that the data is completely separate from each other.
Oh, sorry.
Getting back to the reproduction question, we have also seen nudibranch egg cases. I believe that might be in the highlights video. Very lacy, beautiful looking egg cases, and an enormous number of whelk egg towers with whelks actively laying eggs. Just enormous aggregation of that on one of our past ships.
We had the question if anyone knew why they build the towers in that shape or had any guesses or ideas.
I don't know. I'll look it up.
It's interesting that the whelk itself is, a spiral shape, and they lay their eggs in a spiral, creating the tower. It's just kind of a neat thing.
I don't know this for sure, but it may be that just naturally, I've seen several gastropod egg cases. They do come out in a spiral form. So it may just be part of how they do it. But in terms of getting it up into the water column, I'm thinking that if it is above the sediment, maybe it gets more, it keeps it clean a little longer, perhaps, with current passing by. I'm not sure if that's true or not, but I would guess that that might help.
It's a great job there, Jake.
We have a hagfish swimming left. And a tanner crab.
Our new bearing is 110.
There's a nice gate up in the middle of the lasers.
It will be slightly downslope at this heading.
Tom said it was a long
So
nose
you and
gate.
Dan. (110), yep. If you're finding that you're getting too much blue, then you could crab a bit.
So I have a question for Taylor Ann. Typically on the ship, at the end of each dive, we have a crew of people helping getting all the samples off the ROV. But with our limited number of people that can be on the ship, I noticed her doing it pretty much alone the other night. Is that true? Were you able to keep up?
Yeah, so there's 3 of us on the science team. So we were processing samples last night. The reason why you saw me alone is because I'm also collecting samples for my master's thesis research. And I was filtering some microplastic samples. And yeah, I just didn't want to keep everyone up waiting on my water samples. So I told them to go to bed and I just finished up. But we did process all of the biological samples and some ocean acidification samples and eDNA samples together. And it went by pretty quickly. We got, I think, what was it? Maybe 18 or maybe 15 samples where we processed them all pretty, like I think maybe within 2 hours.
Argus, you're heading still at 045.
So yeah, and then we have people that help out too that aren't on the science team. So sometimes the chief scientist, Nicole, will come and help, and some people on the communications team, and everybody likes to see what the samples we're bringing up. So if they have the free time to be involved, then they are welcome to be if they're aboard the ship.
As scientists on shore, we missed that part, is actually seeing the specimens when they do come up.
Yeah.
Scientists, just to interject here, we are about 200 meters away from our last end if we wanted to do a transect. Otherwise, we're happy to just keep trucking along as is.
Yeah, let's do a transect. We have the opportunity and we have some good conditions, it seems.
Okay. Pilots, you all set up? Looks like it. So let's, Taylor Ann, we'll start the next one.
All right, that will be transect 12.
Roger. Yeah, so one of the reasons why we were talking about the heading being slightly up and not direct is because we're going a bit kind of down slope-ish. So we want to make sure that Argus isn't upslope from Herc too much, otherwise he can't drop his delta. So it's kind of a bit of a game. This is not too bad of a slope either way. But if it was a bit steeper of a slope and we had to do something like this, we might do that crab along.
We have a couple more elementary schools tuning in. So I wanted to say a quick shout out to Tincture, 6th grade, so middle school for Tincture. And then we have Lafayette Elementary School tuning in. Hello to the 5th graders.
Hello, 5th graders.
Welcome.
So glad you're able to join us.
With this world of telepresence and doing school at home, it's a great opportunity for students to tune in.
Taylor Ann, if you're able to, could you tell us a little bit about your microplastics sampling?
Sure. 1 second, just notating what I'm seeing here. Yeah,
Yeah,
so...
if it's not a good time, don't worry.
Oh, no, you're fine. So I'm collecting microplastic samples here in the deep sea to kind of get a profile of what is here. So we're trying to see the different types of microplastics that are present in the deep sea across various depths associated with different water mass properties. So as you get deeper and deeper in the water column, the properties of the water change, and I want to see how the types and concentrations of microplastics will change according to those water mass properties. So microplastics are ubiquitous, which means they're everywhere in marine systems. And sadly, we're even seeing them in the deep sea, even though you'd think it'd be pretty hard to find a little piece of plastic all the way at the bottom. You do see it. And there have been some studies in Monterey Bay in the past that have found microplastics in the deep sea. So part of my research from my master's is in the deep sea here in the Monterey Bay National Marine Sanctuary, and then we'll be moving on to the Channel Islands area. So I'll be collecting some Niskin samples, so water samples, to see what microplastics are floating in the water, as well as some biological samples. So we collected some drift kelp to see if there would be microplastics on that. And also be collecting some sea pens, which feed in the water column, and possibly some holothurians that feed in the sediment to see if they are ingesting microplastics from the sediments. So the second part of my research, which is why I'm interested in the kelp on the seafloor, is actually going to be in the kelp forests of Palos Verdes, the peninsula in Southern California. And I'll be seeing if kelp forests act as a hotspot or kind of a sponge for microplastics because of the physical environment of the kelp forest. So the thick densities of kelp in kelp forest actually changes the way that water flows and slows down water. It makes it kind of sit in one place for a long period of time. So my hypothesis is that we could be seeing some microplastics accumulating there because there's less water movement. So I would be interested in seeing if the kelp that we found on the deep sea floor would have microplastics on it too, and if that can kind of transfer from one ecosystem all the way down to the deep sea. So nobody has really looked at that yet in terms of giant kelp. So we'll see. I'll let you guys know what my results are in the next year.
Fantastic.
Thank you so much for that
sharing.
summary.
Yeah, of course.
We're getting geographic shout outs from all over the world and the country. We've got some hellos from Scotland, from Tennessee, from Alabama, Germany. People are tuning in from all over.
I know we had Norway on the other night. So a minute ago or so, you might have seen something kind of rolling along the seafloor. That was a market squid, the tiny squid that probably in the last stages of its life. We saw one just before this dive, just kind of hanging in there, but usually they are way up in the water column and not happy when they are rolling around on the seafloor bottom.
What
We
are
are
we seeing
indeed on
here,
transect,
Kirsten?
yeah. But you're doing fine. You kept a decent speed.
We have
Looks like another Tanner crab.
A Tanner crab.
Yeah.
Also had the question of, if you see something interesting, can you stop and look at it when you're on transect?
We could, it's not preferred because we do have to modify the data. So it's better to just keep going. But if we do see something really interesting that we haven't seen before or we want to collect, we can always drop what we call a waypoint. It marks where it is in terms of latitude and longitude, and then go back to visit it. So we do like to just keep going because these transects are standardized in terms of length, time, and speed. And so interrupting that sort of makes us have to modify how we analyze the data.
So we've just
But I think
passed
after this
over,
transect, we'll do a little some exploring.
we just passed over a small juvenile thorny head down on the right. And then there was a long spine. And I didn't quite catch what that next thorny head was. It's like a couple of Dover sole.
Had an appreciation from another deep sea researcher, amazed that Nautilus's operations are up and running and able during this time to continue with their planned missions. And mostly wanted to say, how concerned or what protocols do they have to kind of maintain the links and the internet connection? Because that's the part that she's most impressed with. Anyone want to take that or have any thoughts on that?
Guessing Steve's over there.
Yeah, well, we have a dedicated satellite internet access set up. It's interesting, the satellites that we use were also being used by wildfire firefighters on the West Coast over the past month or more. So that actually limited our bandwidth a little bit and was limiting the resolution of our feeds to shore. But we are, I believe, back up to our full bandwidth and bringing you the crystal clear images hopefully you're seeing at home. But it's not, yeah, it's not a common service, the one that we subscribe to. It's expensive, and we do it so we can have this telepresence, which is one of the biggest goals of Nautilus and OET, is to bring all our exploration live to anybody who is interested.
It really is an amazing gift for OET to provide this to the public in this way. I think once people get turned on to watching a dive, maybe in their area, they get turned on to the ability to watch Nautilus live anywhere, wherever it is in the world, which is why we have so many people from around the world tuning in. It's a really unique opportunity. And the fact that this platform was so built out made dovetailing into this pandemic so much more smooth for the scientists to be able to. Sorry about that. I had connection difficulties for a moment. We are just going to pass a long spine thorny head. You can see the third dorsal spine is significantly longer than the others.
I'm seeing a few snails in here. Maybe we'll get some more egg cases coming up if there's some hard structure.
Well, Nautilus, we have fans from Florida, Santa Cruz, Ontario, Canada, Columbus, Ohio. Norway wanted to let us know they are here today as well. and Namibia. So getting a lot of folks chiming in with whether listening or watching.
That's great.
And just to add to that question about how we maintain our connection, the satellite dish that we have on board the ship needs to stay pointed in a very precise direction towards the satellites it's communicating with. And to do that, there is a very sophisticated gimbal system which corrects itself many times a second and keeps everything stable for the connection while the ship is rocking every which direction in the seas.
I think my mic might have been off. Justin, did you hear me do my shout out back to Norway?
We did not. Now's your moment.
Okay, just in case those folks in Norway happen to be my family, say a shout out to Ellen and Anton and Noah. Thanks so much for joining.
I know we're all in the moment. We're being very present with what we're doing right now, but we do have a question of when is Davidson Seamount happening? Erica, do you want to take that? Are you free for a moment?
Sure. So at our daily briefing, I got an update on our schedule. We will be ending this dive later tonight. And then tomorrow,
Sorry.
we will be transiting to...
This crab claw.
Crab is back.
Yeah.
Tomorrow we'll be transiting to Davidson, and we hope to be on site tomorrow around 4 o'clock Pacific Daylight Time. And we're going to try and get a full day in. I don't know the number of hours yet, but we may be experiencing some tough weather come Sunday morning. So we'll try to dive between tomorrow evening and early Sunday morning. So keep a lookout on the Nautilus Live website to see what our schedule will be exactly. But we are keeping track of the weather. It's going to get a little windy and the swell is going to come up a little bit. So some of that high weather limits getting the ROVs in the water.
Okay, we're just at 150 meters there, so we can mark the end of transect.
Thank you.
T12E.
So we have some time to explore as we head out onto our next waypoint, or the direction we're going. So if anyone in the science team has any requests for looking at maybe some of these snails or burrowing holes, if we get to a fish, they might scatter away, but we might see a crab we might want to get a close up of. So
Speaking of crabs, could we check out the front porch while we're off transect and see if our Tanner crab is right below us?
Yeah, if you look at the satellite V3, you can also see the Herc brow cam.
There's our Tanner crab. So this crab has joined us at noon today. So it's been with us for an hour and 45 minutes. Maybe we can make sure to give it a gentle nudge before we leave the seafloor.
This is a ride of a lifetime here. This crab is never going to get this opportunity again.
I thought it was volunteering to help with sampling later.
2 extra arms.
Crab's like, ungrateful humans. I helped out on the 2018 cruise, and they don't even remember me. I mean, are they even? I'm sure I molted once or twice, but come on.
All right.
Actually, now that we can see the carapace, this is the same crab that we were looking at that Jennifer was talking about. The tube worms had set up homes on the carapace and legs of this crab. And this is actually the same individual. So those little white lines on the legs and back, so the pattern is the same as the one that she was talking about.
Not that big. Not that big of a crab.
What was that about the markings on the crab? And
yeah, so on the last shift, Jennifer was talking about the on that particular crab, there were some little white lines on the legs and on the back, and those are actually worm casings, homes that some worms had set up. So she was pointing out the fact that these crabs molt regularly to be able to grow in size. They molt their shells, inflate themselves when they're in a soft stage, and then harden their shells again. And the fact that those worms had been able to settle out and set up on the carapace just meant that made it look like it had been a little while since this crab had molted.
oh, cool.
Just anecdotal.
If anybody wants to keep track of the crab, it's on satellite feed 3. You can see those worms.
I just want to mention that there is a significant market for tanner crabs and the related snow crab, which is not the one we just saw, the snow crab. And they're commonly caught by benthic trawl, and they're sold at markets in Alaska. And female tanner crabs can produce over 86,000 eggs. So they're quite productive. And they brood their eggs between July and September, and the brood duration is about 11 months.
Oh, I think
They tip.
What's going on? Do we have a topside?
I think it's the topside.
Topside reset?
Yeah. All right. I was wondering why I was drifting up all of a sudden. Oh, don't leave.
And the species we're looking at is the grooved tanner crab, Chionectes tanneri. Thanks, Kurt.
So last night at midnight, so I let you guys sort things out. So following up on the capabilities of the ROV, there are specimen boxes that are in a tray underneath us right now. And last night on the midnight to 4 a.m. shift, we collected a couple of beautiful large coral specimens. And those specimens, those are long-lived species, and these were large individuals. They were collected to do some aging and to look at, to have, create a kind of a climate story, a ocean climate story from the information that's left in the skeleton. So
yeah, he drifted south, so he's trying to get back.
Yeah, I was just, there we are.
110.
So we really try and limit the samples that we collect to the things that the information that we specifically need, but that's really important data. And there will be a highlight coming up on the Karina fish who that bamboo coral specimen was collected for. She's going to be highlighted in a video on, what was that, tomorrow? Is that the 9th?
Yeah, I think so, the
It's
9th.
the 9th, yes. Mm-hmm.
Talking about her work with bamboo coral and aging. And all of the important information that comes from that.
Jake, do you want me to zoom in past all this stuff? Or do you need to straighten out first or anything?
Yeah.
This is the first shift, this cruise that we've had the interaction with
Drifting a bit
folks
south.
being able to chat with us. And it's really fun. So thanks, folks, for reaching out. It's nice to know that people are out there and they're curious and interested.
And thanks,
Fun
Justin,
for us.
for playing the SCF role today.
Thank you so much, Justin. Yeah.
My pleasure. I was busy looking up when Karina and
Oh, you
Steve's
had the
talk
auto out
is going
funniness.
to be.
Yeah.
Because it restarted.
That's going to be on Friday, October 9th at 1 p.m. Pacific Daylight Time. So you can go to nautiluslive.org and look at the education events. And there's a link where you can watch Karina Fish and Dr. Steve Oskovich's talk about corals in the dark.
Our very own Steve.
So I'd like to keep an eye out for another Umbellula sea pen. We collected one the other day, but upon retrieval, it had drifted out of the bio box, so we lost that sample. So keep an eye out. I just saw one earlier. We'll probably see some more.
There is a sea pen, but does not look like the right species.
I'm sure we'll find one.
You guys can reset the crab camera if you need it for flying. Okay, cool.
The snails seem to be the common animal around here, with some crabs and Dover sole intermixed in.
Erica, did you see the Jan's comment on the sample?
I did. So we're not going to make Umbellula a priority.
It's good job, Jake, getting back there. Still a bit south, but making headway. Argus's heading is south, so that's why
Yeah, it looks.
you're heading out. Yeah, good job.
Justin, feel free to stick with us as long as you like.
Thank you. I was just touching base with Amity about that. I'd like to stay on for a little while. I'm having really enjoying being with you all, feeding you guys questions. I asked you this, Kirsten. I never got a response. Do you actually have the feed of questions? Or are you just that intuitive? Because you often answer them before I've had a chance to feed them to you, which makes me wonder if I'm, is my job as important as I feel like it is?
No, I do not have the feed, and we all greatly appreciate your presence.
She's just a mind reader.
It's kind of creepy.
Maybe.
Speaking of kind of creepy, did we just see another hagfish go by?
We did.
Yes, we did. I have to say, on my last ROV cruise on the Shimada, we were looking at this kind of mass of hagfish, and several of them were curled up tightly into a really tight little spiral. And we zoomed in on them, and I was slightly grossed out a little bit, and then went into the mess hall, and there was this like tray of cinnamon buns. I don't think I've been able to eat a cinnamon bun ever since.
We have two species of hagfish off our coast, the black hagfish and the Pacific hagfish. And the black hagfish tends to occur between 600 and 1200 meters. We're currently at 720 meters, so we're most likely seeing that. But I wouldn't rule out seeing the Pacific, but most likely we're seeing the black. And they feed on a variety of organisms. They eat squid, octopuses, shrimps, fish parts, polychaete worms, amphipods, eggs, and euphausids. But they're pretty well known for feeding on other dead things like whale falls. And they'll actually try and get inside the animal and eat from the inside out. And they sometimes emit slime to deter other predators in the area. And that'll actually clog the gills of other competitors, making it more the hagfish able to be a better competitor for eating the carrion.
So we are currently at 719 meters. So we've come up significantly over the course of this dive. I think we landed at about at around midnight at about, what was it, 950 meters? This is probably the shallowest dive. Is that right, Erica?
So far, this cruise. Mm-hmm.
So scientists, we're probably at a pretty good deciding point here if we want to head downslope to 2U or head to kind of a long contour to 2V.
Let's head towards 2V.
Okay. Yeah, it's looking like this is indeed artifact. We're not seeing any change in seafloor here.
All right, thanks for that.
Sure.
If you like maps and navigation, you can check out channel 3, which has the contours and the boat location, and that's fun to follow along. Some people might not be in quad view and see that.
Okay, I'm going to adjust course to 090, which should be approximately a long contour.
Router.
Bridge nav. I'd like to change course to 090, maintain speed 0.3, and we can do 150 meters steps. Thank you.
And if you're having any trouble reading that map on the quad view, satellite feed 3 does have its own stream. You check out for